Welcome to The Explainer. Today, we're dissecting a specific SBN biology trial paper question to uncover exactly how your body maintains its internal balance, and we'll be looking at how your urinary system acts as the ultimate filtration plant. Let's just dive right in.
So, here's our game plan. First, decoding SPM question 5. Then, regulating carbon dioxide, followed by the principles of homeostasis. From there, we'll hit temperature and blood sugar, the structure of the kidney, and wrap up with the exact process of urine formation.
All right, Section 1, Decoding SPM Question 5. I want you to think about this for a second. How does your body actually detect hidden internal changes? I mean, how does the brain know when the internal environment shifts? Like, say, when your blood becomes just a little too acidic? Well, the secret lies in specialized sensory receptors. These little guys are incredible. They literally convert these hidden chemical shifts into electrical nerve impulses.
Take a look at receptor X, right here, sitting securely in the medulla oblongata. This is the central chemoreceptor. Its entire job, its precise function, is to detect changes in the concentration of hydrogen ions, which means it's constantly tracking the pH value in your blood and cerebral spinal fluid. It's essentially your body's built-in, highly sensitive chemical alarm system.
Moving into Section 2, regulating carbon dioxide.
So imagine you're doing some really vigorous activity. Right away, the partial pressure of carbon dioxide in your blood shoots up. That CO2 dissolves to form carbonic acid, which then breaks down. Suddenly, you've got this massive spike in hydrogen ions, and your blood pH drops. It becomes acidic. This acidic shift is what stimulates those central chemoreceptors we just talked about. They immediately fire off a nerve impulse directly to the respiratory control center in the medulla oblongata, telling your body, hey, increase that breathing rate.
Now, when you compare how the body regulates carbon dioxide with how it regulates blood pressure, this really cool universal rule in biology emerges. Even though the triggers are totally different, the core mechanics are exactly the same. Both rely on a negative feedback mechanism. Both are trying to wrangle that internal environment back to normal. Both kick off with changes detected by specific receptors, both involve the transmission of nerve impulses, and both route those critical impulses straight to the medulla oblongata. The players change, but the game is exactly the same. Which brings us to Section 3, Principles of Homeostasis. Let's zoom out a bit. The textbook definition of homeostasis is the regulation of physical and chemical factors of the internal environment within normal ranges for the cell to function in optimum conditions. But why does that actually matter to us? Well, if physical factors like temperature or chemical factors like blood sugar and pH go too far off script, the enzymes running your cellular metabolism will either slow down completely or denature. Homeostasis is literally what keeps your cells alive when the outside world gets chaotic.
Let's jump to Section 4, Temperature and Blood Sugar.
First up, blood sugar. Your pancreas is the absolute hero here, running a negative feedback loop to keep things right in that sweet spot, usually 75 to 110 milligrams per 100 milliliters. When blood sugar gets too high, beta cells in the pancreatic Langerhans cells secrete insulin. Insulin swoops in, stimulating your muscle cells to use that glucose and converting the leftovers into glycogen to stash in the liver. But on the flip side, if your blood sugar plummets, alpha cells step up. They secrete glucagon, which signals the liver to convert that stored glycogen right back into glucose. It is a perfect biochemical seesaw.
Now, what about temperature? Thermoreceptors in your skin and hypothalamus are constantly monitoring. If you get too hot, your physical responses go to work. Vasodilation widens the blood vessels so more blood flows near the skin surface to radiate heat. You start sweating to cool down, and your erector muscles relax so your hair just lies flat. But if you're freezing, the exact opposite happens. Vasoconstriction pulls blood away from the skin, your skeletal muscles start contracting rapidly, that shivering, to generate heat, and those erector muscles pull tight, raising the fine hairs on your arms to trap a nice, insulating layer of air. Next up, Section 5, Structure of the Kidney. The human kidney is an absolute marvel of biological engineering, and it really comes down to two major functions. First is excretion, which is essentially taking out the trash, clearing out toxic nitrogenous wastes like urea, uric acid, ammonia, and creatinine. The second is osmoregulation. This is all about balancing the total volume of water, the concentration of ions, and your blood osmotic pressure. Your kidneys are constantly fine-tuning the chemistry of your body fluids behind the scenes. And to pull this off, the kidney uses millions of these microscopic functional units called nephrons. It's an incredible journey.
It starts at the Bowman's capsule, which wraps around a super high-pressure cluster of capillaries known as the glomerulus. From there, the fluid makes its way through the twisting proximal convoluted tubule, dives deep into the U-shaped loop of Henle, climbs back up through the distal convoluted tubule, and finally empties into the collecting duct. This winding, continuous pathway is exactly how fluid transforms into urine.
Bringing us to our final section, Section 6, the urine formation process. So how does the nephron actually filter blood? It happens in 3 distinct sequential phases. It all kicks off with ultrafiltration, where high hydrostatic pressure literally forces fluid out of the glomerulus and straight into the Bowman's capsule to create glomerular filtrate. As this filtrate moves along the tubule, we hit phase 2, reabsorption. Your body is incredibly meticulous here. It saves exactly what it needs, actively pulling 100% of glucose and amino acids right back into the blood, with water naturally following via osmosis. Finally, we get to secretion. This is where waste materials, things like excess hydrogen ions, potassium, and toxins that didn't get filtered the first time around, are actively pushed from the blood directly into the distal convoluted tubule. Whatever is left by the time you reach the very end of the collecting duct, that's your urine. We have covered a tremendous amount of ground today, from the central chemoreceptors hiding out in your brain all the way down to the microscopic nephrons hard at work in your kidneys. But I want to leave you with one final provocative question. What happens to the body's internal environment when these crucial negative feedback mechanisms completely fail? Think about it. When the pancreas simply cannot produce insulin, or when the nephrons are severely damaged by high blood pressure. Grasping this perfect balance of homeostasis is really the very first step to understanding massive systemic failures like diabetes or kidney disease. I really hope this explainer has brought these complex concepts into sharp focus for you. Keep learning, keep questioning, and I will catch you in the next explainer.